Welcome to Pharmacy Prep. In this video, I like to discuss about Cytochrome P450 drug interactions. Cytochrome P450 enzymes are very important enzymes that are responsible for many drug interactions. So there are major drug metabolizing enzymes are CYP450, which is 73% of distribution of these enzymes, and then UGT, which is urinyl diphosphate for glucuronidation, which is 15%, esterases 9%, and monoamine oxidases about 1%, and then other small enzymes. Well, here's the distribution of those. The major enzymes for drug metabolism is CYP450 enzymes. So what exactly this cytochrome P450 are? These are the family of heme-containing monooxygenases type of enzymes. That means, as the name tells you, monooxygenases means that's basically it is responsible for oxidative reactions. So they catalyze the wide range of oxidative reactions. So there is again within those cytochrome P450, there are sub-enzymes. Among them, the most common enzyme is CYP3A4 enzyme, which is approximately 34%. And the second most common enzyme is CYP2D6, which is a 22% of the subtype of enzyme. Well, sometimes you wonder how did this P450 number come from? Basically, this P450, which is due to their maximum absorbance at 450 nanometers, that is a pigment of 450 nanometers when it bound to the carbon monoxide. So therefore, based on that 450 nanometer pigment of that protein, that's what those P450 comes from. Otherwise, there are approximately 30 type of major subtype of enzymes of CYP enzymes. Among them, CYP3A4, 2D6, 2C19, and also 2C9, 1A2, and 2B6. Those are the commonly seen with that responsible for various drug interactions. Well, to understand those drug interactions, we need to have some simple basics of drug metabolism. So once drug is ingested through the oral or taken by the parenteral routes, drug is distributes into the various organs. And from those, drug undergoes two phases of metabolic reaction. One is a phase one. Phase one metabolic reaction primarily takes place in the liver. Liver is a major site of the cytochrome enzyme metabolic reactions. So the cytochromes, obviously, they do oxidize the most of the drugs, and then phase two metabolism, generally referred as detoxification pathway, because they undergoes conjugation reaction. That means they attach to those glucose and sulfates and acetyl groups, etc. As a result of that, those conjugated drugs in the phase two metabolic reaction, they become water soluble. And once they become water soluble, eventually that helps drugs to excrete from the body. And also sometimes some drugs, they undergo same time phase one and two reaction and produce reactive metabolites. Those reactive metabolites some of them can result into toxicities, and then obviously some of them can result into conjugation and obviously excretion from the body. Well, this is the summary of phase one reactions that include other than CYP enzymes, mainly include the other reactions or combine all reactions. The phase one reactions are known as functional group reactions, and then phase two are conjugations. However, in this video, my primary focus is cytochrome enzyme drug interactions. So to understand the cytochrome drug interaction, it is very important to understand there are three actions of the drugs that happens on the cytochrome enzymes. I mean, when drugs bind to those enzymes, they produce three types of effects. One is substrate, and then they do inhibitors, and then there are inducers. So let's look at what exactly substrate is. Substrate is a drug when it binds to those active sites of the enzyme. So here, let me give you more details about those active sites. So example is CYP enzyme, active sites. There are some sites in the enzymes that are responsible for drugs binding to them and producing metabolic reactions. Those sites are known as active sites. When a drug binds to those active sites and undergoes formation of a new metabolites, and that is what exactly the substrates they do. Substrates are the drugs, they bind to those active sites of the CYP enzymes and they form those new metabolites. And then next we have the most important thing here is inhibitors. Basically, inhibitors are the drugs, they bind to those enzyme sites. There are two sites here. One is the active site, and then other site is allosteric site, which is other than active sites. So drugs can bind to both either active site or allosteric site, and eventually they inhibit the or they decrease the activity of the enzymes. So that is what referred to the inhibitory effect. So when these drugs they reduce the activity of this enzyme, eventually drugs metabolism stops or decreases significantly. As a result of that, drugs will not metabolize and they stay in the body for a longer period of time, and eventually that can result into toxic side effects of the drugs due to the inhibitory effect. And then next we have inducers. Inducers, they function very similar to inhibitors. However, they bind to those active site or allosteric site. They increase the activity of the enzyme. So as a result of that, when you increase the activity of the enzymes and drugs get metabolized faster and also decrease the pharmacological effect of the drugs. So now these three effects happens when drugs bind to those enzymes. Well, when a person takes more than one drug, eventually some drugs they may undergo as a substrate, and some of them can function as an inhibitor, or some of them can function as inducers. Eventually, that lead to the drug interactions. So therefore, let's go into details on those substrates and inhibitors and the types of mechanism of those inhibitory effects. Before that, I like to discuss about some basic things of those enzyme activity that is called substrate binding affinity, which is referred as Michaelis Menten constant. Well, before going to the Michaelis Menten constant, I like to give you briefly understanding on binding affinity. So obviously, enzyme has an active site. When the drug bind to those enzyme, how strongly that bind to those site. Like the binding affinity is the enzyme strength or attraction to the substrate, like how strongly bind to it. So obviously based on those binding affinity, the substrates can be classified as a weak substrate, intermediate type of substrate, and the strong substrates. So here is a question alerts. So binding affinity, which is a concentration at which 50% of maximum metabolic reaction occurs. So just to let you know, for the exam purpose, you have to be more focused on the strong affinity substrate because they results into more serious drug interactions. So often questions in the exams are more focused on the strong substrates. Let's talk about a little more details on the Michaelis Menten constant. So this can be explained by a Michaelis-Menten plot. Here on the y-axis, the reaction, the enzyme activity or reaction velocity, and then x-axis is the substrate concentration, or you can say drug concentration. So once you take a drug and increase the concentration, initially the reaction velocity increases and reaches to the maximum velocity of those reaction, that is known as Vmax. So once it reaches to the Vmax, then it reaches to the saturation. So that means once it reaches to the saturation, even you increase the substrate concentration, that maximum velocity will not change. So now let's talk about how do you relate that Michaelis-Menten constant Km. So basically you take half of the Vmax is equals to the Km for this drug. So now let's say this drug is drug A, and now you compare with another drug, that is a drug B. So the drug B here, the blue line, the Km of the drug B is here. So therefore, there's a difference in the um, affinity of drug A and drug B. So that means by using this concept, you can say the drugs with higher affinity have a less binding, means decrease in the binding capacity, binding uh, affinity. So therefore, Km is inversely proportional to binding. So that means the drug B here, the blue color, has the higher Km. That means that has a less binding onto the site of the active site. All right, so here is a question can be possible in the exam. So what is the purpose of the Michaelis-Menten constant? Michaelis-Menten constant uses the substrate binding affinity is measured by using Michaelis-Menten constant. So that's the purpose use of this Michaelis-Menten equation. However, we also have other various clinical, the experimental use of the Michaelis-Menten formula, which is very important one, very famous concept. Well, let's talk about the another important concept that is important to understand whenever we talk about the drug and drug interactions. That is called intrinsic clearance. Intrinsic clearance measures the ability of liver to clear the unbound drug, because that means the liver is the major important site of the drug metabolism. So how much drug is cleared from the liver, that is known as intrinsic clearance, which is not related to blood flow and binding considerations. So there's a very important formula here is intrinsic clearance is inversely proportional to the Km. That means higher the when the drug has a less Km or decrease in binding capacity, eventually lead to the higher intrinsic clearance, and it will be inverse of that as well. Like if you have a lower the intrinsic clearance, higher the binding capacity. So that concept of those intrinsic clearance is plays a very important role in most exams. That's a big in the exams. All right, so what is the big in the exams is CYP450 inhibition mechanism. Well, there are CYP enzyme inhibition divided into two types. One is a reversible and irreversible. So further reversible, further divided into competitive and also non-competitive reversible enzyme inhibition. So let's talk about first the reversible competitive inhibition. So competitive inhibition is, for example, a drug enzyme active sites. Here, the drug A and drug B. Those two drugs are competing for the same active sites in the enzyme. So imagine when you're competing those two drugs for the same active site, eventually competitive inhibition compete for the site. That means the one that has a higher affinity to those active site will bind to those active site. For example, here drug A has a higher affinity, therefore it binds to the active site. And then whereas drug B is unable to bind. So there is a one terminology often used in the literature, that is a drug A here is a perpetrator, whereas the drug B is a victim. Well, that means is in this case, the victim drug have the basically have the Km, increase in the Km, and then eventually lead to the decrease in intrinsic clearance. So low intrinsic clearance means there is an increased risk of adverse drug reaction. So one thing important about is the competitive inhibitor, which is a very frequent question in the exam, and also clinically plays a very important role in clinical practice to related to drug interactions. So to minimize the competitive inhibitions, two competing substrate, if you use concomitantly, it can result into drug interaction. To avoid the drug interaction, you can space time interval almost equal to 5 half-lives of time intervals of the drug that helps to reduce those drug interactions. So let's say if a drug has a 10 hours half-life, so we are talking about 50 hours, which is nearly 2 days time. So the other drug have to be at least used after 2 days to minimize those drug interactions. Next, competitive inhibitors also, example here, just take a CYP theophylline, which is a narrow therapeutic index drug, which is well known for serious drug interactions, and often questions in the exam from the narrow therapeutic index drugs. That's a big for the exams. All right, so now let's talk about theophylline, which is the substrate of 1A2, and then eventually it does bind to them. However, when you concomitantly use with another drug that is duloxetine, and duloxetine has a higher affinity to 1A2 compared to theophylline, eventually it does displace or take off the theophylline, and theophylline will remain in the blood circulation, eventually lead to the decrease in the clearance. So as a result of that, it increased the adverse drug reaction of the theophylline. So that's a very potential drug interaction can result due to the combination of theophylline and also the duloxetine. All right, so next we talk about the competitive inhibition, also referred as reversible, because competitive inhibition is reversible because it is also depends on the substrate concentration. Means how much when you have two drugs a person is taking together, if one drug concentration like depends on their half-life, if a drug has a higher concentration, even though that has a low affinity, eventually displaces the drug from the active site. For example, let's take example here, a drug and a drug A and drug B. So let's say drug A is a yellow color here, and now increase the concentration of the drug A. Eventually, drug A displaces the red color, which is a drug B, and bind to those active site. So that is a basically reversible because of substrate concentration. Or if when you decrease this concentration of the drug A, that is yellow color, eventually then drug B will bind to those active sites. So that kind of reversible competitive inhibition is a very common in clinical drug interactions. Well, very important point here for the exam is the question alert is the high concentration of weaker affinity substrate can displace even though stronger affinity because it is a concentration sensitive substrate concentration sensitive. That's a very unique, very frequent question about competitive inhibition, which is reversible. Even you increase the concentration of one of the drug compared to other drug, then that means you have a higher dose or higher concentration in the blood circulation during that period. Eventually, the drug with the less concentration will not bind, will remain in higher, and it cause results into drug interaction. So next we talk about the non-competitive inhibition. In the non-competitive inhibition, important interesting point here is one more important terminology you have to know. There's a sites other than active site, there are other sites where the drugs they bind, that is also known as non-active site, which is also known as allosteric sites. So when drug bind to those allosteric sites, so eventually that can influence the active site or modify their conformations. So for example, when allosteric site a drug binds to them, and eventually when it modifies the conformation of the active site, and now the drug that cannot bind to the active site because now this site of the active site structure conformation is modified. So that results into non-competitive inhibition. So what is the biggest difference and what is the key point that you have to remember for the exam here is non-competitive inhibition cannot be reversed or by cannot be changed by overcome by increasing the substrate concentration. That's the biggest difference between previous competitive inhibition and non-competitive. This is not reversible by increasing the substrate concentration. That means one have to wait to get drug completely out of the blood till to avoid any drug interactions. Here is an example of non-competitive inhibitions. For example, enzyme 2C9, which is a substrate of carvedilol, whereas the fluconazole inhibit the 2C9. Fluconazole bind to those allosteric site and then modifies the active active site conformation. As a result of that, carvedilol cannot bind to the um, CYP2C9 enzyme. Eventually, that results into accumulation of carvedilol. As a result of that, when someone takes fluconazole and carvedilol combined together, that can result into increased side effect of carvedilol, which can result into hypotension and also can increase the side effect bradycardia. So let's look at the next one is the next, we look at the combination of competitive and non-competitive. There is another type of enzyme inhibition that is known as mixed inhibition. So the mixed inhibition is a good example can be a ketoconazole, which is a strong, potent CYP3A4 inhibitor. And so midazolam is a CYP3A4 substrate. So mixed inhibitors are simply basically why it is called mixed because they do bind, they also compete on the active site and also they compete at allosteric site. As a result of that, that is known as mixed inhibition. For example, ketoconazole that is bind compete on the active site with the midazolam and since it is a potent and has a higher affinity, it bind to those active site. As a result of that, metazolam will accumulate in the blood circulation, can result into sedation. Additionally, ketoconazole also bind to the allosteric site. So therefore, that can result into changes into the conformation of the active sites. As a result of that, midazolam again will not bind, eventually lead to the higher sedation side effect of midazolam. So generally, mixed inhibitors are usually more potent than competitive inhibitors. All right, finally we have the CYP enzyme inhibition that is a irreversible and mechanism based inhibition. They are much more interesting. So there are irreversible and mechanism based inhibitions are two types. One is the alternate substrate inhibition. So here is an example of this is a CYP3A4 enzyme. The classical, the most common example of the drug interaction and very serious drug interactions is clarithromycin. Clarithromycin is a strong CYP3A4 inhibitor. So basically, clarithromycin binds to those active sites. After binding to those active sites, it forms this stable intermediate that is known as nitrosoalkene intermediate. And that nitrosoalkene intermediate make those covalent bonds onto the active sites. As a result of that, that is a makes irreversible inhibition. And Also, it results into serious drug interaction with many substrates. The second type of irreversible inhibition is the suicide inhibition. This also has a classical example, CYP2C19 enzyme with the substrate, the inhibitor that is the omeprazole. Omeprazole is a classical example, or proton pump inhibitors. Many of them are example of CYP2C19 enzyme inhibitors. And that makes also basically this these drugs, they bind to those active site and they destroy the enzyme structure by binding to them, and that make them irreversible suicide inhibition. So the classical drug interaction with this is a clopidogrel. Clopidogrel is an antiplatelet drug, so if someone, and also clopidogrel is a substrate of 2C19. That means clopidogrel is a prodrug. It has to become active drug. To become active drug, it has to be required 2C19. Whereas if someone take concomitantly together with omeprazole and clopidogrel, clopidogrel will not metabolize because omeprazole is completely bound to those CYP2C19 active site and eventually leading to the destruction of that enzyme. And now there is clopidogrel does not have any 2C19 for further its metabolism. As a result of that, lead to the drug interaction. All right, so hope this information was useful, and stay tuned for more drug interaction and more videos, and subscribe Pharmacy Prep channel. Thank you.